Hello, everyone. Welcome to part 2 of chapter 8. In this section of the chapter, we're going to discuss the phenomena of graded and action potentials. Um, Buckle up. There is a lot going on in this video, much less the entire chapter. It's a big chapter, and this is a big video. I've got 43 slides. Hopefully we can move through some of them quicker than others, try to keep the time down, but I don't want to sacrifice explanation and context on your behalf. So strap in and let's hope for the best. In previous lectures, we've talked about resting membrane potential, but what we're doing now is we're causing variable membrane potential. We're causing changes that are dynamic, not only in terms of their value, their overall magnitude, but also over time. So we have graded potentials, and that means that they can be different. Just like you can get different grades depending on how you do on an exam, you can get different kinds of graded potentials. So we're going to look at where they are, where they occur in the neuron, and how they're created, and then this concept of summation. That is to say, things add up. Just like you were using an abacus or something, you're going to add and subtract to come up with a summation. Then we have action potentials, which are of a similar phenomenon, but they have very, very different characteristics. And one of the things we're going to do is illustrate the similarities, but we're also going to emphasize the differences. One of those major differences is that in the action potential, you're using a voltage-gated sodium channel that actually has two gates. So we're going to go through that mechanism in detail, partly because this channel is exceptionally important in a pharmacological context. It's very important for anesthesia, and in some cases, heart drugs go after the sodium channel in the heart. It's going to be a detailed mechanism of action here. We're going to talk about refractory period because what this does, this refractory period gives us an entry into how action potentials can create differential signal. I haven't explained that yet, but it's a way of creating emphasis or de-emphasis on a particular set of signals coming down an axon. That's about the best way I can put it. We'll talk a little bit about saltatory conduction, myelination, and getting to velocity, which is our planned lab for this week. Again, it's going to be a virtual lab. We're going to give you some data and then have you calculate velocities and see the difference and so forth along those lines. Love to do that lab in person, but till we get to that time, we'll go with the virtual lab. Then lastly, just at the end of the slide, we'll talk about how disorders of ion concentrations can lead to pathologies of neurologic function. We're going to start first with graded potentials, and in order to start with graded potentials, I'd like to show you these images of something creating waves on a smooth surface of water. That's in a way how graded potentials work. There's an area of depolarization or hyperpolarization in some cases, and that causes a spread of depolarization or change of voltage across the surface and through the neuron cell. But you know the waves are taller here than they are here. That's because there's only a given amount of energy imparted into the system at that point. And so as the waves get larger and larger in radius, the energy has to spread over a larger and larger area. And that's somewhat analogous to what we see with graded potentials, is that they may start off strong, but as they move further and further away from the origin of the stimulatory signal, I should say, the deflection, the amplitude of voltage gets lower and lower. And we'll explain reasons why. Here is that phenomenon illustrated graphically in two ways. So here is the stimulus point of origin. This is a depolarizing stimulus. We're using sodium in this case. And so the voltage rises the most at that point of origin, but as you spread away from that point of origin along the cell surface, the membrane surface, the overall amplitude of voltage change weakens. And then that's shown here. They're showing the numbers here to correspond with the numbers here and or here. So they have a big deflection of voltage here. This would be a positive deflection. But as that sodium spreads and the voltage moves away from this part of the cell, it gets weaker and weaker as it goes, just like that droplet of water in the previous video. Great figure question for understanding this idea that we have a diminishing intensity of depolarization from the point of stimulus. And that's something that action potentials do not do. They do not lose their strength. The typical way that graded potentials are created is by a change in permeability to an ion in a given spot on the surface of a neural cell or a muscle cell membrane. What happens, like for instance, let's say this is sodium channel here. It opens up and sodium will want to flow in to the intracellular space by virtue of both the chemical concentration gradient as well as the electrical charge. This is a negative on this side, it's positive on this side. So the sodiums are going to flow in, but they're also going to spread out as they move towards those areas of negative charge. Membrane potentials are very much a local phenomenon in and around the membrane. Yeah, things can move through the middle of the cell, through the cytoplasm, but generally what you're going to see is a wave-like phenomenon along the surface of the membrane. So the sodium rushes in and it begins to spread, and you can see it's got a diminishing concentration over area as the area increases. Okay, and that's part of the diminution of the voltage deflection. This flow of sodiums or ions, whatever ones are moving in here, we call local current flow. That will be particularly important when we get to action potentials. Both graded and action potentials utilize local current flow, but action potentials are more dependent on it, especially in myelinated axons. But we'll just point that term out now so that you can kind of visualize that it's the spreading of the positives along that inner surface towards the negative charge that's localized along the surface. Graded potentials are graded. What that is to say, they can vary in amplitude and they can vary in being depolarizing or polarizing, I should say, because we have repolarization and hyperpolarization. Potentials can either be a depolarizing stimulus or a polarizing stimulus, and they reflect stimulus strength. The stronger the signal, the more amplitude in that graded potential you have. It's a wave of depolarization if it is depolarization, or it's a wave of polarization if it happens to increase the polarity of the membrane. In either case, it's spreading through the cell. It's moving through the cell. And they lose strength. We've already illustrated that. They lose strength as they move through the cell due to a couple of things. One is what we call current leak, and I'll explain that here in a second. That's a little more complex than it might seem. Yes, the spreading causes a diminution of the wave, but these are actually even more so weakening that, so it actually accelerates the diminution. The other one is what we call cytoplasmic resistance, and that's basically a diffusional type of resistance. So yes, the sodiums want to move along that inner surface of the membrane, but it's still a sort of form of diffusion after a while. And so these are kind of the two major forces alongside that just increasing radius, if you will. And I kind of want to describe these a little bit more. If the signal is strong enough, or the combination of signals, the summation of signals is strong enough, they will reach this trigger zone in the axon-hillock initial segment of the axon and trigger an action potential or set of action potentials down the axon. So the decision point is whatever the strength of that initial graded potential is once it gets right here. As I mentioned, graded potentials can vary in strength. They can reflect stimulus strength, but they can also be depolarizing or polarizing. Again, the difference between repolarizing and hyperpolarizing is in reference to the -70. If they're going from greater than -70 to -70, would be repolarizing. And if they're going beyond -70, it'd be hyperpolarizing. But in both cases, it's inhibitory. And so here's this figure. I've shown you this before. So any stimulus that causes the voltage to rise is excitatory. Any stimulus that causes the voltage to get more negative is inhibitory. Now we have this concept of threshold, and in neurons the typical threshold voltage is -55. So if we hit -55 in that axon hillock initial segment, it'll trigger one or more action potentials. If we push the voltage away from, in this direction away from threshold, we're preventing it and or making it harder for it to reach threshold. If we're pushing it towards threshold, we're making it easier and or possible to reach threshold. But that's -55 is the typical threshold voltage. And that's the voltage required at the axon hillock initial segment to fire off one or more action potentials. Right here. If we hit that, then you're going to see the action potential. We'll talk about that in a little bit. One major characteristic of graded potentials is that they can summate. That is to say, their influence on membrane potential can add up or be subtracted from the net voltage, if you will. Now we have three different neurons interfacing with this target cell here, this target neuron. A and B are both excitatory neural terminals. That means whatever they're doing, they're triggering entry of cations into the intracellular space. That's why they're considered excitatory. EPSP, excitatory postsynaptic potential. C over here, the channel that's sensitive to its neurotransmitter is letting in anions, or perhaps slowing down cations. I don't want to get too crazy, but let's just say it's letting in anions. It's causing the voltage to get lower. That means it would be an inhibitory postsynaptic potential. In either case, these things can add up. And they can add up and lead to a final voltage down here as to whether or not we fire off an action potential. But we're going to start with the simplest of scenarios, and let's just say A is the one influencing the cell by itself at the moment. So A can fire off, and then the graded potential dies and goes back to resting potential, current leak, that sort of thing, or cytoplasmic resistance. So it might be pretty robust, but by the time it gets here, it's not strong enough. And then we fire it off again. The same story. Gets a little bit of deflection, but we haven't hit threshold. But let's say we get two rapid sequence fires, bing, bing. Well, we've deflected a little bit, and then that second one causes more deflection, and as we're measuring it here, that second deflection has occurred before we could recover, and so we hit threshold and we fire off an action potential right here. We were sub-threshold before because these two different stimuli, both coming from A, were far enough apart temporally that the voltage recovered to resting before it went again. But this one, the second stimulus fired off before we could recover, and so we have that much more deflection. Now we've hit threshold, and that creates a new phenomenon called an action potential. But that's temporal summation. Multiple potentials from the same presynaptic input. It's just a matter of how often they fire off. Do you get summation or not? The other one is, let's say we get a subthreshold from A and B, so they would both look like this, but again, they fire off close enough in time that they create an action potential. We call that spatial summation, like A and B. Or perhaps A and B fire off and they would be enough to create threshold, but C fires off at the same time. It negates some of that, and so we never reach threshold because it's causing voltage to go away from threshold. That's the nature of summation. It can involve temporal inputs and it can involve spatial inputs. Just for a theoretical, I don't have a good picture of this here. Let's just say we take A and put it right here. maybe if we measured right here, we could see a much higher deflection than we see here. But if we put that guy right here, it might be an instantaneous threshold because the strength of the stimulus is enough to fire off an action potential immediately. That's another aspect of spatial. If graded potentials get weaker over distance, then putting a signal right here means it's not going to weaken hardly at all. And it might mean an immediate action potential in that situation. And this is just sort of illustrating some of those points that I've made in the previous slides. If we have three measurement points, all measuring at the same time, we get our stimulus here, and you can see that it's quite strong. It gets us to -40, well above threshold, but remember, we're measuring here. This isn't the axon hillock. That signal has to travel either along the cell membrane or through the cell, whatever way. Along the way, wherever we measure it, we see that we're just barely at threshold right in the middle of the cell body. You can see that it's delayed in time. It's taken a millisecond or so for that wave to get there. And then when we measure down here, again, it took another millisecond to get to that next distance. Now we have weakened to the point it's sub-threshold. It's not going to fire off an action potential. If we have a stronger signal, or if the signal is closer to the trigger zone, yeah, we've got a nice robust signal here, more than adequate if it were at the trigger zone, but it's not. And it's weakening, it's still above threshold here, but we make to threshold, and we should show an action potential deflection. If we had another probe, you'd see that big action potential spike, because we get a big old depolarization during action potentials. But in this situation, we were at threshold when we reached the trigger zone, so we're going to see one or more action potentials fire off. This is a really lovely chart for showing you the similarities and differences between graded potentials and action potentials. It's a wonderful reference. I don't want to go through everything because, again, I want to try to limit my time, but this is a great place to double check that you understand the similarities and differences in graded potentials and action potentials. If you can't find it in the book, then you can certainly find it in the PowerPoint handout. The only little change I would make is that I would probably keep potassium out here in this one as well. It's a minor one, but nevertheless, it can play a role. In action potentials, it's only sodium and potassium. In graded potentials, sodium, chloride, calcium, and yeah, to some degree, potassium. Okay, that's the only change I would make here, but you can read through these and just read through them one at a time, or use this to double check your understanding. Good chart. And now on to action potentials, a complex phenomenon, very, very important from a pharmacological and certainly a neurological standpoint. So we're going to drill into this mechanism pretty well. Action potentials have a couple of key characteristics that I want to point out. Is that they are the long distance in the signaling in neurons. They are self-reinforcing. That is to say, they don't lose magnitude over time, or they shouldn't. They are relatively high speed, though the speed does vary. The diameter of the neuron, whether or not the neuron is myelinated, affects the overall velocity of the action potential wave. But action potentials are all or none. When you hit threshold, you should fire off an action potential. If you don't hit threshold, you shouldn't fire off an action potential. A little bit of a tricky figure here. What they're showing you here is a cascade of dominoes falling, and that's all well and good. But what we have to remember is that over time, as this wave spreads, it's not changing in amplitude, and that's the aspect of the dominoes here. But these dominoes should begin to stand up again, because what happens is you do have a reset going on. But this one right here at 1, it has fired off, it has returned to voltage, it has recovered, and now it's back at basal. It's ready to fire off, probably ready to fire off again. So it should be standing up, if you will. This figure here is showing you 11 electrodes all recording over the same period of time. And so it's recorded a wave. And so this one is deflected and it's recovering. This one's a little bit earlier in the recovery phase, a little earlier, a little earlier, a little earlier, and so forth. It's showing this over time, and it's showing that wave that has spread. If you want to do an experiment at home, if you have a piece of cord or a piece of rope, stretch that rope out between 2 points, like maybe have 2 people hold it, and then give it a flick. You'll see that wave moving, and think of that wave as the voltage on any area of the neuron. Maybe I can find a GIF, and I'll put that in the next slide. It just occurred to me, and I don't want to re-record this video. But it's trying to show you that wave-like phenomenon of depolarization and repolarization. And so I found a GIF for you. And again, think of the rope as the voltage on any patch of that axon. Rather than this being the axon itself, it's showing you the voltage along the axon. And so what you see is a deflection that's moving in a wave-like form, and that's basically what's happening in an action potential. And here we go with the big mech for action potential. We've got 9 steps here. You should know what's going on at all 9 places. Use the figure to your advantage. I would give you this figure or a figure like it and ask you to tell me what's going on in different places. Like for instance, at 1, we're at resting membrane potential. At 2, this patch of membrane, and it's showing only one spot on the neuron. It is not showing any other spots. This is a single point on which an action potential is passing through, shall we say. So at 2, we receive some kind of depolarizing stimulus. Usually it's in the form of local current flow from another patch of membrane. So something has depolarized a patch of membrane, those ions move into this patch and start depolarizing it. If there is enough depolarizing signal, we hit threshold, and that ensues 4 through 9. At 3, when we hit threshold, channels open up, and these are sodium channels. It's a voltage-gated sodium channel, so when the voltage is sufficient to trigger that first gate, sodium flows in. The membrane becomes permeable, increased permeability to sodium. As sodium flows in, it really depolarizes the membrane. We go all the way to plus 30. We shot right past 0, we hit plus 30. At around plus 30 or so, we close a gate. We actually close a second gate. So when we're over here, one gate is open and one gate is closed. The gate opening here is called the activation gate. The gate that's closing here is called the inactivation gate. But I'm getting a little bit ahead of myself. The permeability drops. Okay, the permeability drops. As the permeability drops, another channel opens, and this is a slower opening channel. It's a potassium channel. So the increase in permeability of potassium, but potassium is flowing out of the cell. And so it's a cation, and as it flows out of the cell, you're losing positive charge. And so potassium starts flowing out of the cell, and we start rapidly losing charge. This is the repolarization phase, or the falling phase, if you will. And that channel stays open, and we actually shoot past the resting membrane potential. We actually have undershoot here. We're actually in a phase where we are hyperpolarized. Now, when we hit about minus 80 or so, or minus 70 or 75, those channels start closing, and so we bottom out around minus 80. And the sodium-potassium ATPase is doing its thing, so it's pumping potassium out of the cell and sodium in, and we recover. So we open the activation gate, we close the inactivation gate, we open the potassium, we close the potassium, and then we recover from this deviation back to resting membrane potential. So rising phase, falling phase, undershoot, and recovery to resting membrane potential. But the channels are doing something all the time, and so we have to add that layer of complexity to it. I have mixed feelings about this figure because it's showing you overall voltage and it's showing you relative permeability of each ion. What it's trying to do is showing you that during the rising phase, it's sodium permeability that's going on. And we peak out and then we close those inactivation gates and the sodium permeability falls. Meanwhile, potassium permeability rises, it peaks out during the falling phase, and then it falls, and then we have undershoot over here, and we're finally recovering at the end. It's just showing you permeability relative to voltage. It's a little bit tricky to wrap your brain around if you're not experienced with it. Take your time. It's showing permeability versus voltage. It's basically a graphical equivalent of the GHK or the equation, if you will, on the membrane. And here is one of the major stories, and this is one of the key mechanistic features of an action potential. It is this double-gated voltage channel. A double-gated voltage channel. Notice that it's closed right here, but we also have this ball and chain. So when we are in resting state, pre-stimulus state, the activation gate, the open and closing jaws of this is closed. And the inactivation gate, the ball and chain here, is open. When we hit threshold, the activation gate opens and sodium is free to diffuse into the cell, depolarizing it. When we hit a certain voltage, the inactivation gate closes. Notice that the activation gate is still open. It is still open and the inactivation gate is closed, so we're not letting sodium anymore. What happens is during undershoot, during that hyperpolarized state, we reset the channel so that the activation gate is closed and the inactivation gate is open. That leads to a period of time where you can't reopen this channel. That period of time is known as a refractory period. It means that the channel is not receptive to additional signal. But I'm going to get there. Again, I'm trying to bring these layers on in a conscientious way. Potassium, on the other hand, is just a single gate. It opens when the voltage is right, and then it closes when the voltage goes different. So it's a much simpler scenario. It only has one gate, whereas the sodium has the activation gate and the inactivation gate. Here is the Silverthorne image. It's showing a voltage indicator for the activation gate here, but I think it's missing the voltage indicator for this one too, right? It's a voltage sensitive gate, so why doesn't it have a little plus minus thing? Come on, Silverthorne. In any case, here's activation gate. It's going to open somewhere around minus 55, or, if it's stronger, maybe it's a little bit, whatever. It's going to open as soon as we hit threshold. The inactivation gate is going to close when we hit somewhere around plus 30. So we hit that depolarizing stimulus. Usually that's in the form of local current flow. It causes the opening of the activation gate. Notice that the inactivation gate is still open. This is the rising phase. So sodium is going to flow in and it's going to cause further depolarization of the membrane. This is a positive feedback cycle, this depolarization. So we get those first gates open. Sodium enters the cell and is going to cause more depolarization, which causes more channels to open, more sodium, more channels, more sodium, more channels. The intervening step is the closing of the inactivation gate. So when we get to around plus 30, the outside influence that closes this channel, and you can see figure 810, it closes this channel is the closing of that other gate. Otherwise, it's a positive reinforcing cycle. During the rising phase, we hit peak, we start to close out an activation gate. Meanwhile, those potassium channels have been taking their sweet time opening. By the time we hit around peak, we have enough open that you get a substantial loss of potassium and a substantial repolarization event going on. Once you get this figure, it's a nice way to see action potentials rising and falling. So we're rising here, we're rising, and when we hit plus 30, At plus 30, boom, we close that inactivation gate. So permeability falls. Permeability falls. Now, if we didn't open potassium, what we would see is a nice slow return to resting membrane potential. It would look like a slopey curve as the cell pumps out sodium and as that sodium spreads locally in terms of local current flow. It would be kind of like a graded potential in that way. But we close that and we're losing potassium simultaneously. And that potassium accelerates the voltage loss. But when we hit here, when we get to about minus 70, minus 75, what's going on is that the channel begins resetting. We close the activation gate and we open the inactivation gate. During this sort of recovery phase, during the undershoot region of that action potential. And this is just showing this in another way. So we start with 1 in the resting state, look at the channels, and again, that's a little trickier because you see the gates here as these little flanges, got the potassium channel here and that sort of a thing. But again, I want to always show you multiple figures if it'll make more sense to you. You're going in a clockwise fashion here. I really love this figure, though I know that it is a challenge to take in, especially if you're taking it in from the 10,000 foot level, if you know what I mean. It's actually several graphs superimposed on the same x-axis, which is time in milliseconds. It's a wonderful point of reference, and the easier it is for you to interpret this graph, the better it will be for you, especially down the line if you get into advanced neurophysiology. So we're going to break this up into its pieces. So everything is on the same x-axis here. At the top, we're showing channel status. And at resting membrane potential here at minus 70, our activation little gate, the one that's on the top there, is closed, and our inactivation gate ball and chain is open. Both channels are closed at this point, and the potassium channels are not well illustrated here. I would love to see those two purple ovals close together. When we have enough stimulus that we hit threshold, when we have enough, enough outside stimulus, that could be sodium ions or cations from somewhere coming in, depolarizing us. When we hit threshold, that causes the activation gate to open. All right, so sodium's going to flow in. We got that positive feedback cycle going on. We have a rising phase, and when we hit our plus 30, that's when the inactivation gates close. Look at the channels now. That little ball and chain has moved into the slot. It's plugging the hole. And so at the same time, though, potassium channels are now open. And so now we're in the falling phase. We fall for a period of time, and then we undershoot. And right about at 2 milliseconds, as we begin to recover, the potassium channels are closed at this point. It doesn't show it very well in this figure again. But in any case, we're starting to reset here. Here to here, what we see going on, first of all, right at the beginning, the sodium channels are sort of indisposed. And the potassium channels are still kind of doing their thing. But nevertheless, as we proceed through time, you can see this sodium channel resetting. The activation gate slowly closes, and the inactivation gate slowly opens, until we get back to resting membrane potential. This sort of in-between period is why you don't see, is what is explained or is further reinforced, I should say, down here at the bottom. This bottom is showing the relative excitability of this neuron. Can it fire off an action potential? When we're at resting membrane potential right up until we hit threshold, that excitability is high. As soon as we hit threshold and we're opening up these sodium channels, we go to 0. This period of time during rising and falling phase, there is no way to send another action potential through here, no matter how strong the signal is from wherever it's coming from. That's because we're in what is known as the absolute refractory period. The channels are already engaged. They can't engage any more than they already are. They can't open any more than they already are or close any more than they already are. And when the inactivation gate closed, it can't reopen right away. So there is 0 additional excitability here. But right here at about 2 milliseconds, and this is during the undershoot phase, we begin to increase in the possibility of firing off another action potential. But it starts off really low. It would take a really strong signal to fire off. But as we get further and further, closer to 4 milliseconds, we approach resting membrane potential again, then we're back to high. That's because during this relative refractory period, the channels are resetting, and you can see that. You see that, imagine this is the same channel shown over time. What we see going on is that this channel is changing its configuration back to its initial state that we saw over here. It is resetting. And it's not just one channel in the membrane, there are thousands of them. So as the channels reset, it gets easier and easier for the cell to fire off again. But it is still not high, which means that it's got to be a stronger signal that fires it off. That's because there are channels that haven't reset, and we are still, if I could draw, imagine the yellow as the -70 over here, we're still hyperpolarized. If we were hyperpolarized at all, it would take a stronger signal to get us to threshold. But we're hyperpolarized, and we only have a partial population of channels available to fire off. That's what the relative refractory period is. And that's where the magic begins. Why am I going to all this trouble we're 5 minutes into this slide. Why to go at all this trouble? Because if it was completely absolute and then back to high again, the soonest that another action potential could fire would be roughly 4 milliseconds. We could only fire off bing, bing, bing. But with this relative refractory period, we can now, if the signal is stronger, we can have repetitive action potentials that occur earlier. So instead of a bing, 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 we might be going bing, 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 bing. The rate of action potentials goes up. Or if it's a little bit of a weaker overall signal, bing, bing, bing. Maybe it's at 3 milliseconds or 3.5 milliseconds. We can vary the frequency of action potentials to reflect the relative strength of the depolarizing signal. So we can use an all or none phenomenon to reflect signal strength by varying the frequency of that all or none depolarization. That's the magic, and that's why I spent 6.5 minutes on this slide, probably the longest I've spent on any slide all semester. And I tell you what, I am going to stop right here. We're at 36 minutes. I went long the first time. I'm going a little long here. I'm going to stop on this chart again. Can you compare what I talked about with these two? That's a wonderful way to study. Look at the similarities. Again, remember to write potassium in here as well. And see the differences. Note what I've talked about. The more you can incorporate what I've talked about, the easier the exam will be to refer to, or when you're referring to your notes, it'll take you less time, be less of a challenge for you. So thanks again, and I'll pick up with part 3 and finish off this PowerPoint.